We are really lucky to have our next two speakers speak with us today. So first up, we'll be hearing from Associate Professor Lewis Nacul, who is a professor in epidemiology and public health at the London School of Hygiene and Tropical Medicine. Hope you're enjoying Geelong so far. As well as expertise in the areas of public health, epidemiology, and in general practice and medicine. He's director of the CureME research team and the CFS Bio-Bank in the UK, and last year was appointed to the NIC guidelines used by the NHS. So we're very lucky to have you talk. Thank you, Dr. Nacul.
Good afternoon. Thank you very much for the invitation to be here today. I'm overwhelmed with your hospitality. I'm overwhelmed with the quality of the presentations before mine. So this presentation, I will focus on our experience at the UKME CFS Biobank, but I'm also going to say a little bit about our own research, and I'm going to tell you a little bit about how the infrastructure provided by the Biobank and our research both contribute to translation of science into better health outcomes. That's the face of our research. You can see the top row. These are our researchers from the clinical research department, where I sit, and also Eliana, who is going to be presenting tomorrow. Our expertise is in clinical research, public health and epidemiology, and also we look after the biobank. The lower column shows our heart scientists, and they provide the expertise in immunology, genetics, gene expression, genomics, and virology. It's a relatively small group, but we have many collaborators within and beyond the London School of Hygiene and Tropical Medicine. When I talk about translational science, what I mean is the long pathway between basic science and patient benefit. I'm going to come back to this slide, but the chap there in the corner, where is the laser here, represents a person with ME CFS. And he's going to be present in other slides. You're going to see him or her again. But the location here is it should be in the center of the slide. But the reason he's here, or she, is because they are very interested in everything that happens, all research that we've been doing. But at the end of the day, they want to get better. That's the main agenda for people with ME, and we should not forget that this is the main focus or the main reason why we do research. Right, so let me move to Biobank, which is the focus of this presentation. So Biobank is a repository that stores and manages biological samples for use in research. That's the definition. So Biobank is not only about safely and securely storing biological samples, such as blood, for example, and other samples long-term, but it's very important there is good quality clinical data linked to it, and also it's important that these samples are there for a reason, and they are, of course, the reason is to do research, but also very importantly for sharing. So why biobanks? What do we need biobanks? Why are they useful? They provide a very reliable source of data and samples with high standards of quality and quality assurance procedures. And they are cost-effective and time-effective. So say Paul Fisher wants to replicate their findings with a sample of 100 people, maybe 200 people with ME/CFS, and say you want to compare with controls, both healthy controls and disease controls. Of course, if you think about the amount of time that it's going to take you and the resources it's going to take you to recruit all these patients, to characterize them, to get blood samples, etc., time is ticking and the resources are going. So you can alternatively obtain samples from a biobank with good clinical data attached to it, and of course your work will be, you have speed in your work and you save resources. And of course the type of research can be done with biobanks are many, and these are just some examples of the sort of things that we've been discussing here today: formulating and testing new hypotheses, understanding disease pathways, investigating disease subgroups, and biomarker discovery. So let me tell you about the UKME Biobank. This is, for short, is UKMEB. It's actually not located in the London School, or at the London School. It's located at Royal Free Hospital, University College Hospital Biobank. And the reason for that, we are just 20 minutes apart. The reason for that is because they already have a state-of-the-art biobank which serves many research groups. So they are 24/7 attended, so they are fully alarmed. So they have all the rigorous high quality procedures to keep a biobank. So our biobank, the UKMEB, is actually nested within the Royal Free Biobank. It's a very cost-effective way of having a biobank. Right, we started now many years ago. I think it was in 2011 when we started discussing the idea of setting up this biobank, and we did it in a very in partnership using a consultation process which involved people with ME, their carers, very extensively, and also involved researchers, clinicians, and experts in various fields, including in human tissue banks, of course, in law and ethics. Contribution of people with ME was essential in the process. So these are just example quotes from people with ME when we're discussing the setting up of the biobank. They found it would be very important to ensure quality and linking to relevant data. That's what they told us. For example, if we do it, the samples need to be of very good quality and have the appropriate information attached. And because we don't know what area is going to go down, we have to be quite broad in the information. And that's what we did. We have quite comprehensive clinical information, other information attached to each individual time point. And they had some concerns. This is another example of a quote from a patient, a person with ME. If you're trying to get as much people as you can, they are afraid what you're going to do, whether the government would get hold of it, whether the insurance companies could use it, or whether benefit agents could use it. So they are concerned. When they share the samples, when they share the data, they want to be sure that we treat the data and the samples appropriately. And this brings us the importance of high ethical standards. So, of course, we need to maximize the benefits of the biobank, but at the same time, we need to be very aware of the importance and our responsibility of the type of samples and data that we're keeping. And we are guardians of the biobank. So, of course, we don't want anything to be misused. So this is a list of all the ethical procedures we went through before setting the biobank. I'm not going to go through each of them. I just want to point out that we hold a Human Tissue Authority, HTI license. This is happens not only the London School has HTA, but also, of course, the Royal Free Hospital Biobank has an HTA license. So it's fully qualified to hold a biobank. And of course, we have informed consent and free withdrawal. So far, of the about 600 participants, we had only one person who decided to withdraw from the biobank. And all samples, of course, are de-identified. So who are our cases are people with confirmed ME CFS, and he used the following criteria: the CCC, CDC 94, or CUDA, and the previously known Institute of Medicine, or the SAID criteria. And in addition to, so these are the criteria used for recruitment purposes. All our patients at recruitment are between 18 and 60 years of age, and they have different degrees of severity of ME/CFS, including about 25% of them are actually severely affected, which we define by being mostly bedbound, but certainly housebound. And these are the patients that we visit at home and collect all the samples and clinical information through home visits. We have two control groups. One is the population control group of healthy people, whatever healthy means. And the other group is a disease control group of people with multiple sclerosis. And these are group matched by age, sex, and area of residence. So that's the samples that we obtain. For each individual time point, we have on average 46 aliquots of blood derivatives that are generated. And these include whole blood, serum plasma, red blood cells, PBMCs, and blood for RNA. And as it March this year, we have just over 30,000 aliquots from patients and controls. In total, we have about 600 participants. About half of them have just one assessment. About half of them have at least 2 assessments. And by the end of next year, some of them will have 5 follow-up assessments with an interval of around 6 months between the assessments. So we have longitudinal data, which is very important also for researchers, as mentioned today. Right, so what sort of data we have? Just some examples of clinical data. So of course, we have anthropometric measurements. We have blood pressure lying down and then standing up, and then up to 10 minutes standing up. We have spirometry. We do this is the hand grip. This device is used to measure the hand grip power. So we give to the participants the hand grip, and they produce a maximum force, three times with an interval of half a minute between them. And each force is up to 5 seconds. So these are examples of the clinical data that we have, and we have comprehensive data from questionnaires, including from the UKMEB Symptoms Assessments questionnaire, which enables to confirm the diagnosis according to the research criteria that we use. And then we have the participant questionnaire. This one is a more comprehensive questionnaire for those who are admitted into the study, and they provide comprehensive clinical information on symptoms, clinical history, as well as socio-demographic, family history, and data on potential exposures and before the illness started. And then we use validated questionnaires such as the SF-36 for quality of life outcomes, and then fatigue scales such as the fatigue severity scales and analog scales, and also pain scales, and the GHQ-28 for psychological morbidity. Right, very importantly, the Biobank has been serving, of course, our own research, but this is a resource for sharing. So we use an open doors policy. Anyone can apply for samples, including academic and commercial users. And we have some priority areas, but which are listed there, but pretty much the samples are available for any ethically designed study. So what are the procedures if you want to obtain samples and data from the Biobank? You use a one-page outline application, just giving you an idea what the research is about. And this is quickly revised by our steering group scientific subcommittee. And then usually being approved, we ask for a full application. People who already have obtained, were subjected to peer review because they wrote a grant application and got funds, then we we don't necessarily need to review again, of course. But it's very important that people have ethical approvals for their own studies. The Biobank gives us ethical authority to share the sample, to store and to share the samples with researchers internationally. However, the specific research that people are doing, the research that is being conducted, requires specific research, which is the responsibility for the researcher to obtain. And until this is done, we will not release the samples. This is a very important part of the procedure. And of course, once all is done, then we complete MTAs and DTAs, and we provide aliquots of the relevant blood derivatives to the requesting researchers, accompanied by the identified data. Right, so far we have shared samples, or we are in discussion with researchers actually across the globe. We have South America, North America, and of course we have quite a few in the UK and other places in Europe. The Middle East, represented there, it's by Israel, and then Australia. We have one sharing of our data and samples with this country, I'm pleased to say. And we want more requests from you. We really have lots of aliquots to distribute, and we want good research to be done with the biosamples that we store. Right. Let me talk about translation a little bit. So I showed this slide. I'm showing again. So the biobanks are very useful for the first steps of the translation path. Basic science in particular. We also provide potential individuals for clinical trials. You may wish to select people for clinical trials on certain characteristics. You can consult our database, and then you could potentially develop clinical trials, of course, depending on further consent with our participants. But there are lots of steps beyond that. We still need good further studies beyond initial studies of basic science, initial clinical trials, to be able to accumulate good evidence, which can be translated into clinical practice and then eventually be translated into better health for individuals and for the public with ME/CFS, for groups of people with ME/CFS. So it's quite a lengthy process there, and there are many impediments. There are many things that can go wrong during this translation pathway. So, and these are very unfortunate because these impediments may prevent biomedical science from realizing its potential, which is to be useful for the public. In this case, to be translated into better health outcomes for people with ME. But we know that we're trying to address the second point there, of particular obstacles, including the access to well-characterized biological materials in biobanks. So we hope we're doing our part by having the UKME Biobank. And it's interesting that it has been reported that the risks of failing to translate the science are perceived to be mainly due to delays in providing diagnostics and clinical care to patients. And if you ask someone with ME/CFS what they want, what they think the research should tell them, what answers they want is, we want to be diagnosed, we want diagnostic biomarkers, and we want to have good clinical care. So I think this, I'm sure, will be familiar to you, these ideas. So this is the Translational Science Values of Death. I didn't create this name, but I think it's there just to highlight the fact that many people can, many things can go wrong since the discovery phase or the basic science and on. So there are lots of good basic science research studies which actually don't move forward into the clinical stages. And many good clinical trials will not feed the evidence base and will not be put in practice. And if you don't overcome those valleys, if you like, will not reach the ultimate outcome of translational research, which is health practice, good health practice with good health outcomes. And there is a problem that we should not be jumping from one valley to the other necessarily. So let's suppose the situation where we have bad evidence practice and you try to jump the bad evidence into health practice. This may this have a potential to harm people. So we need to be very careful in the way that we mount evidence and we apply it in clinical practice. So it's quite a lengthy process. You need to be aware of the impediments. Right, let me tell you a little bit about our research from CureME. So we are a multidisciplinary team or group of people. We take very seriously participatory research. Participatory meaning with the full participation of people with ME. And I'm just going to mention very briefly some of the studies that we have done. For example, I'll show you data in a moment about the health impact of ME, having ME on disability. I'm going to send you some results about our hand grip strength use in people with ME compared to other groups of people. And when you measure blood pressure and heart rate variation, we are in a position to facilitate subgrouping of people according to autonomic dysfunction, for example. So I really like simple techniques. So because I'm a clinician and I see patients, I really think it's very important that we can apply simple techniques to diagnose people with ME and then to provide whatever support they need. And one of the simplest ways to provide this is by examining people, looking at them, and measuring their blood pressure, for example, asking them to do a spirometry, getting their saturation of oxygen. Those are just examples of the sort of data that we collect information on, or having the hand grip. So I think this is, of course, simple technology that have a potential for translating very, very quickly into practice if you find something useful with these. And one of them, of course, we go through routine laboratory tests, and one of them is, for example, the CK, and the CK is creatinine phosphokinase, as you know, is the main energy provider by muscles, as well as the heart and the brain. And of course, we haven't published yet, so I'll just briefly mention that we found the CK levels are substantially reduced in people with ME, particularly severe ME cases compared to controls. And this is after adjusting and controlling for activity levels. Of course, CK are very dependent on activity. So this is a test that we routinely use. I think most groups of clinicians will request this test. And if we have values below a certain cutoff point, which is about 50, we have a sensitivity of over 96% to actually pick up a case of severe ME. It's not very specific, but it's very sensitive. So this is just an example of a simple test that we can use, and I use on a day-to-day my day-to-day dealing with patients. My colleagues do immunology research, and we also, still unpublished, but it should be published hopefully in the next few days. We look at NK cells, phenotype and function. I must say that we didn't find as florid abnormalities in NK cells as many of our colleagues in other groups have found. However, we found clear abnormalities in people who actually have CMV infection. And then this will have abnormalities in NK cells. And interestingly, in relation to T cells, we found abnormalities in CD8 positive mate cells, which are a specific type of T cells, which are very relevant for gastrointestinal mucosa defense. So this is something which we hope to be publishing very soon. In relation to genetic expression, we found some similarities in the distribution of, or an expression of genes between actually people with severe ME and people with MS. That's interesting. And of course, there are some further research and that are in progress. Let me show just a few concrete examples of data that has been published already. So I just mentioned to you how serious you take patient participation. So they were from the beginning of the process with us, working with us. Our steering committee is chaired by a patient representative. It's not an academic who chairs, it's just a patient representative. And when we had the first results before any publication, we actually had workshops and did some qualitative studies of people with ME and also people with MS to ask them to help us interpret the results. And that's just some quotes from them. One quote said, the fact that Biobank breaks down Canadian criteria, non-Canadian criteria was important. So it's this thing about subgrouping is stratification by criteria. That's something that I found quite useful. And we use this in our reports. And then another thing, the next quote, there is a fairly big distinction between mild, moderate, and severe cases. And from that, it looks like there were two separate conditions. And this is very important because we think about, usually I think, we think about mild cases, mild, moderate, and then severe cases as a continuum of the same disease, of the same problem. But there is always a question, is it actually the same thing, a severe case of ME? Are you talking about the same disease, or are you talking about different diseases, those who have milder forms and those who have very severe forms, which are based mainly bedbound or housebound? And then another interesting quote, we haven't got a magical cure yet, and I don't know how many years it's going to be, but we do have such a fantastic resource that will enable much more research to be done. I think this is very nice to hear, and it's a witness of the recognition the patients find the resource very useful. So this is some evidence about quality of life and how much it is disrupted in ME/CFS. If I show you this, we use SF36 for those who are not familiar. SF36 have 6 domains which refer to physical function and 1 physical component summary, and then we have 4 domains which relate to mental function, and then we have a mental component summary. And these are normalized rates. So normalized rates, the healthy controls have all the indicators between 50 and 60. That's what the normal population have. And then we have people with MS. Of course, they have reduced quality of life. And then we have ME. ME is the center. The more you go inwards, which means lower values for these indicators, the worse off you are. And we found that people with ME in all domains are have significantly lower values than healthy controls, and all but 2 of the indicators, they are significantly lower than MS. And the only ones where they are not significantly lower is in mental health, which is similar, and role emotional. All others, people with ME are more severely affected than people with MS. What's the importance of this? How do you translate that? I think this is very important for validation of our results. Validation validation of the patient disability. So I think it's about legitimizing the disease in one hand, and it could potentially be used by social workers, by planning social support, social services support for people with ME. I think this is just an example of translation potential. So this is another output from our data. So we, as I mentioned, we quite comprehensively phenotype people with ME in relation to symptoms. And let me tell you what this says. Oops. It shows here that healthy controls, and this is physical component summary, so this is about physical well-being, and this is mental component summary, this is about mental well-being. So if you think about a healthy person, the stronger predictor of feeling unwell from the physical point of view is pain. And from for the same healthy controls, sleep dysfunction is the stronger predictor of mental health, or not fitting well from the mental point of view. I'm sure you can relate, those in this room who are healthy, or those who can remember the time they were healthy, I think you can relate. If you have pain, you feel physically bad. If you don't sleep very well, then you tend to have some sort of emotional instability. That's what happens with healthy controls. And see how when you go to multiple sclerosis, the stronger predictors for physical health is neurocognitive symptoms, of course. It's a neurological disease, as ME is. And for mental health is autonomic. And let me remind you, many people with MS have problems such as incontinence, for example, which ends up being classified as autonomic. But let's go to ME, and then there is an important difference between ME, the milder cases, and the more severe cases. For the milder cases, the main predictor of being unwell physically is post-exertion, are the post-exertional symptoms. For the severely affected are actually neuroendocrine symptoms. And in terms of mental well-being, for the mild cases is again sleep dysfunction, similar to people who are healthy. And for the severe is again neuroendocrine symptoms. So what makes people feel unwell or feel disabled is different if you have ME and if you are healthy, or if you have MS, or if you have milder forms or more severe forms of ME. How do you translate that? I think if you're thinking about treatments for different people, maybe we should concentrate on treatments that can address some types of symptoms, depending on the group you're talking about. Of course, until we find a treatment that cures everyone, if we do one day. Right, so it's a final slide about our results, and this is about the hand grip. And we have here, I'm only going to show you this slide. That's the hand grip. They do 3 attempts, and this is how it looks in a healthy person. This is how it looks in a person with chronic fatigue who doesn't have ME. So actually, this is a chronically fatigued individual who doesn't meet the criteria. And then we have MS and milder cases here, and then we have the severely affected here. So you see substantial differences, and there is a trend here as you go from normality to severely affected. So, and there is another interesting thing here, is that when you look at the normality, basically the 3 measures, the first, second, and third, are pretty much the same. There's not much variation. If anything, you do the first one, then you do the second one, it's slightly worse, and then you go for the final one, and then you put a lot of effort, and then you can get it a little bit better results in the last one. But when you look at the severely affected, see what happens. First measurement, second measurement, third measurement, so there's a downward trend there. Is this a positive, or this is 2 minutes? Summary. So just summarizing, just my final slides, summarizing. Just to remind you, we use partnership to establish the biobank. We believe we have robust protocols, and we can facilitate research in very cost-effective ways. We share data and samples and encourage everyone to apply. And of course, we are convinced the biobanks can support research translation, but at the same time, there are many gaps in the translation path that still need to be addressed. And in terms of way forward, we understand there will be a biobank here in Australia, or at least we hope there will be one. And of course, I'd be very keen to collaborate with you guys. At the same, as we've been collaborating in Europe, Eliana is going to tell you more about our European Club network with 22 European countries. So we try to standardize procedures for biobank across Europe, and we also work with SOVE, CFS in the United States to standardize procedures in relation to biobank and data collection internationally. So just to finalize, our guys here again, and what patients need. I just want to, when you think about translation of science, we need to have in mind what patients need, and that's what they tell us. And the patients here can can have different views, but they want diagnosis. They want treatment, they want good services, and they want respect, and they want to be supported. And I'll tell you, even if we don't have a specific treatment, even if we don't have a biomarker for diagnosis just yet, we can still provide diagnostic services from clinicians because we can diagnose clinically. We can still provide we can still help people in terms of management. We can think about services, how to develop service to better serve the patients. And of course, overall, we need to show respect and support to the patients. Research needs to address better diagnosis, biomarker, pathophysiology, stratification of cases, but also, very importantly, I want to highlight that we haven't been hearing much about delivery of services. It's very important. That's the final bit of the translation. We need to have proper services there. And how many countries actually in the world have services for ME which are organized, integrated? Very, very few, I'm sure. So I probably need to stop here. Of course, I just want to mention that the services need to be available and they need to cover the population according to needs. And of course, the severely affected is a specific group that we need to learn better ways to address them. Is telehealth the answer, or can you go to everybody's home? And we need more specialists in ME, and we need to educate the primary care physicians so that understand more about the disease. And this is my acknowledgments one. The main funders at the moment are the ME Association in the UK, a charity, and the NIH in the United States. Thank you.
It's not uncommon to have multiple family members with ME/CFS, and I'm just wondering whether you've managed to recruit any families where there's a high incidence of ME/CFS, particularly from the point of view of GWAS, and as an extension to that, whether you've got any twins in your registry there, and how you're going about trying to recruit those patients.
We don't have twins in our register, but we have family information. So we have information about people who have other members of the family with ME/CFS. But our recruitment procedures, we usually try to avoid blood relatives, and so we tend not to recruit people, relatives with ME, but we have all the information there. So if there is future study that we want to go for family studies, we have the data there and we can look for them.
Is there a reason why you're not taking any blood relatives who also have the same disease? Because that would be an enormously powerful tool in the GWAS type of approach.
We discussed this a lot when we plan, and of course there are advantages and disadvantages. We didn't know how many people we were going to get, so we decided let's have people with independence in relation to family clustering of disease at that stage. But this doesn't mean that we shouldn't perhaps be reconsidering that.
On there in the interest of time. Thank you very much, Dr. Professor
Thank you.
Nacul. Thank you.